Okay, so today we're going to cover vesicle trafficking, and before I get into this, I have to be honest, this is my favorite lecture of the year because I studied vesicle trafficking and cellular transport when I was in graduate school, and it's just something that's near and dear to my heart. I'll point out some of the things I worked with and worked around many of the people who created all of the images and all of the information that's in this lecture. So it's a lot of fun, and I hope you guys enjoy it too. Okay, so cellular transport's an essential process. So cells need this to survive. If they can't move proteins around, then they die. Right? It's just an essential process. So we saw last time co-translational and post-translational translocation into the ER. And so once a secretory protein has entered the ER and it gets properly folded, it needs to get transported to the right location. And we're going to look today really at how the transport is controlled. And it's mainly controlled through small little vesicles, right? Transport throughout the entire cell is mediated by, well, transport through the secretory pathway is mediated by small vesicles. And two mechanisms that you guys need to know is how vesicles bud off a donor compartment and how they fuse with the target compartment. All right, and it's a tightly regulated process, and I'll tell you right up front, it involves small G proteins. Okay. So two things that you need to know, just as a general thing, and I'm sure you've had this in some of your other courses, exocytosis versus endocytosis. So exocytosis is the fusion of a vesicle with the plasma membrane that releases its contents into the outside of the cell, whereas endocytosis is actually the uptake of small molecules into a vesicle that pinches off and buds. Okay, so in exocytosis, a vesicle fuses with the plasma membrane. In endocytosis, is where it buds off the plasma membrane. So this is involved in secretion, and this is involved in uptake. So vesicle transport, or vesicular transport in general, you have a donor compartment and you have a target compartment. Okay, and vesicles bud off the donor compartment, and then they form these small vesicles, and then they fuse with the target compartment. So when they bud, they actually select various proteins, membrane proteins, and soluble secreted cargo. And then when that vesicle fuses with its target compartment, the transmembrane proteins actually become part of that compartment, and it also releases its soluble secreted cargo. Here is all of the secretion and endocytosis that's happening in a mammalian cell. So you have bidirectional transport between the endoplasmic reticulum and the Golgi, and then you have transport between the Golgi and the early endosome, and secretory vesicles and late endosome. So wherever you see a blue and a red arrow, red is going forward, blue is going backwards, and green has to do with the endosome. And you'll see in a second, these are sort of color-coded to help you sort of figure out the pathways. But in general, you have translocation that's occurring at the endoplasmic reticulum, and then small vesicles bud off and go on and sort of fuse and become part of the Golgi, and then proteins then travel through the Golgi, and eventually they get budded off and can go on to fuse with the plasma membrane, or they go on to join up with the early endosome or the late endosome, and eventually that gets transported to the lysosome. Okay, and then you also have secretory vesicles with secreted cargo. Okay, and this bidirectional budding and fusion is tightly regulated by small G proteins. Okay, both the budding and the fusion, as you'll see in a second. Here's an EM of some of the vesicles that cells make. There's the Clathrin-coated vesicle. This was the first one actually discovered. And then there is the COP 1 vesicles, or codomer complex 1. And so these mainly bud off of the Golgi, and then there are COP2 proteins which mainly bud off the endoplasmic reticulum. So my PI, when I was in graduate school, the primary investigator, my mentor, actually discovered these vesicles when he was a postdoc in Randy Sheckman's lab. And this is actually his image, if you want to look at the references in your textbook. So here's where the vesicles form and how they travel. Okay, so you have the COP1s or the COP2s that form at the ER, and they go forward. Okay, so this is considered anterior grade transport, whereas COP1s tend to bud off of the Golgi, and they're largely involved in retrograde transport back to the endoplasmic reticulum. And then Clathrin-coated vesicles tend to bud off and pinch off the plasma membrane, as well as other places sort of later in the secretory pathway. All right. And so this nice color coding actually matches sort of the color coding that you guys saw in the earlier slide. Okay, COP2s, COP1s mainly bud off the Golgi, COP2s mainly bud off the ER, and Clathrin mainly off the plasma membrane in late secretory compartments, as well as sort of the trans-Golgi network. Here's a grade EM of the Clathrin-coated vesicles, and once again they mainly bud off of the plasma membrane and the trans-Golgi compartment, and these were the first vesicles to be identified. Okay. Here's just the electron micrograph of the Clathrin triskelion. This is the major coat protein in Clathrin-coated vesicles. And here's just a little schematic of how it looks. There's a heavy and a light chain. Okay, and these self-associate on the membranes to actually help sort of form this basket-like structure, almost like a soccer ball if you look at it. And so here you can see the individual Clathrin triskelions added here, and that's the vesicle. And there's 12 pentagons and 6 hexagons, and the Clathrin triskelion actually has both this heavy and these light chains. Okay. But the question comes in, where does the Clathrin-coated vesicle know where to assemble? And it has to do with these adapter proteins. Okay, so this is one of those sort of molecular mechanisms that I would hope you guys really sort of focus on and know. Some images like the Clathrin triskelion and stuff like that's important to understand this, but this is really sort of the mechanisms that I want you guys to appreciate. Okay, so if you have the plasma membrane here, and you have a receptor on the plasma membrane, and you can sort of think of this as receptor-mediated endocytosis. And we'll see this again when we're looking at the low-density lipoprotein, the LDL receptor, which when it's mutated, you have actually high cholesterol. But the way it works here is you have your receptor, and it will bind cargo, and the binding of cargo actually attracts these adapter proteins, which can bind to the cargo receptor. Okay, and once the adapter proteins are there, the Clathrin triskelion comes in, and it starts to cause this invagination, and you start to get this sort of vesicle forming. Okay, and we'll see in a second how this gets pinched off. But once it's pinched off, the coat can then be shed, and then you're left with sort of a nascent transport vesicle. Okay. And so this is just one of those mechanisms that, I want you guys to sort of know. And so go back to previous lectures now and sort of see, okay, well, here's like a whole mechanism shown in this slide. I need to know how that works. Okay, that's how you should be preparing for the exam. Okay, so not only are the cargo receptors important for the adapter protein binding, in this case AP2, also phosphorylated lipids, okay, so phosphoinositide PI45 P2, which is here. And so when sort of the cargo receptors and the the phosphoinositides are there, you get binding, and all of a sudden AP2 can come in its sort of open state, and then this will help recruit the Clathrin triskelion. Not shown here is these receptors will typically have cargo associated with them if it's receptor-mediated endocytosis. Okay. So here's another small vesicle. This one actually retrieves acid hydrolases and other things from the endosome, and it's called retromer, and here it is, just a structure of it. I'm less concerned that you guys know exactly how this forms. It just goes to show that there's just many vesicles present in the cell. Okay, and the point is that the lipid composition here is different. Here you have PI3P versus the PI45P2. Okay, so different lipids sort of help to form vesicles on different things because there's different proteins that sort of associate with those lipids. Okay, and just as a general rule, here's sort of all of the different phosphoinositides that can form. Here's just phosphatidylinositol, and then you can get the different carbons all phosphorylated, and they can change, and they'll all be different depending on the cellular compartment and what's budding and what's fusing off. Okay, I'm a little less concerned with you guys memorizing all of these, but I do want you to appreciate that depending on the lipid composition, that actually helps determine what's happening at that membrane. And this is all happening on the inner leaflet of the plasma membrane. And here's just an example of two schematics of how proteins can bind with different phosphoinositides. Okay. And here's just another example. Once again, here's the key over here to tell you where the different phosphoinositides are located in the cell and what's happening. Some are involved in phagocytosis, some are involved in endocytosis, some are involved in regulated exocytosis or constitutive exocytosis. So I don't expect you guys to memorize these. I'm more concerned that you guys are learning the molecular mechanism of, let's say, how a vesicle forms and how a vesicle fuses. Okay, be it Clathrin, COP1, COP2, or whatnot. Okay, so once the adapter protein AP2 has bound to a cargo receptor with cargo, and you have your Clathrin triskelion sort of forming around, and you've got this invaginated vesicle, it pinches off. And one of the protein that helps it pinch off is this protein called dynamin. Okay. And other associated proteins. And it can also bind to phosphatidylinositol, and it also has a GTPase domain, which helps it sort of pinch off. Okay, and once the vesicle has pinched off and it needs to uncoat, oxalin and HSP70 actually help shed the coat, so that then you're just left with the coated vesicle. Okay. And there's actually mutations in dynamin that actually block the pinching off of the vesicle. And here's a perfect example of that. This is a fly nerve cell with a mutation in dynamin, and you can sort of see the Clathrin-coated vesicles are here, but they can't pinch off. They form, or they start to form, they invaginate, but they can't pinch off when you have a mutation in dynamin. Okay, so now we're going to move on to COP2 vesicles. And like I said, this is what I did in graduate school, so I really like it. And this in the next few slides is exactly the type of thing that I want you to know in molecular mechanisms. It's actually a 3-slide sort of thing. Okay, so COP2 vesicles, they bud off the endoplasmic reticulum and they select cargo. Okay, and the way it starts is you have a small G protein called SAR1. And SAR1 is normally in the cytoplasm in a GDP-bound form. Okay, and it has this amphipathic alpha helix that's in sort of this conformation when it's in its GDP-bound form. On the ER membrane, the endoplasmic reticulum, you have the SAR1 GEF. This is the GTP exchange factor, okay? So it's going to catalyze the exchange of GTP for GDP, okay? And in the exchange of GDP for GTP on SAR1, the empapathic helix sticks out and it wants to embed in the membrane, okay? And so this is sort of SAR1 active in the membrane, okay? And so once this has become active in the membrane, it recruits other proteins called Sec23 and 24. These are a complex, Sec23 and 24. And Sec23 binds to the SAR1 GTP, whereas Sec24 helps to recruit cargo via a cargo receptor and cargo. And for those of you who care, my little claim to fame is I actually discovered this protein right here, cargo receptor. There's actually many of them, but one's just sort of shown here. And so this is what I did in graduate school, was sort of figure out how these cargo receptors pull cargo. Okay, so once again, when SAR1 GTP is on the membrane, it recruits Sec23 and 24, and Sec24 actually binds to a cargo receptor, which is bound to cargo. Okay, and then once you have the Sec23 and 24 assembled with SAR1 on the endoplasmic reticulum, then you have Sec1331 come in, and these form a cage-like structure not too dissimilar from what you saw with the Clathrin triskelion. Okay, and sort of the way that happens is you have your SAR1 GTP bound, and that recruits Sec23-24, and that recruits an outer cage structure of Sec1331, and eventually this causes the vesicle to sort of pinch off and bud. Okay, and so once this vesicle has budded off, the question is, how does it uncoat? It has to uncoat to fuse, and so I sort of have this question here, and I'd be asking you guys to answer it, and hopefully one of you would sit there and say, well, the SAR1 hydrolyzes its GTP to GDP. And the answer to that would be yes, that's correct. And the GEF in this whole thing is actually Sec23. So once the vesicle is formed, or GAP I mean, the GTPase activating protein, Sec23 causes hydrolysis of the GTP to GDP, which causes the helix to get removed from the membrane, and the whole vesicle coat uncoats, right? It just falls off. Okay. One note, and it's not in your book, but COP1 vesicles basically form in a very similar fashion to this. They use different set of proteins, but it's very analogous to this. Okay. And so a question I may ask on the exam would be, okay, well, you know, and this would be on your final, not on this next exam, but because it's no longer sort of a question on, you know, would I ask how COP2 vesicles, which your book lays out, I might ask how do COP1 vesicles, and have the analogous protein laid out, something along those lines. You know, I'm trying to give you guys as much information to help you study. I realize this sort of distance learning and sitting in front of a computer listening to me talk can get very boring and your mind can wander, but hopefully you can go back and watch this. But just as a general rule, something like this and like the Clathrin-coated vesicles, know the various steps, okay? How the COP2 vesicles form. They recruit SAR1 GTP. You get the exchange of GDP for GTP. The SAR1 gets embedded in the membrane. That recruits Sec23-24, which then recruits Sec1331. And then Sec23 actually works as the gap to hydrolyze SAR1 GTP, and the vesicle uncoats. Okay. Okay, so now on to fusion. So once the vesicle is uncoated, it needs to go on and fuse. And there's a whole series of Rab proteins. These are, once again, small G proteins that are involved in fusion. And the Rab proteins help direct the vesicle to fuse with the right target compartment. Okay, now normally the RABs, they're just small G proteins, and they're normally associated in the cytosol with these proteins called GDIs, and these are RAB GDP dissociation inhibitors. Okay, these prevent the exchange of GDP for GTP. Okay, but once you have GTP exchange with the RABs, they can actually associate with the membrane. So here's just a whole family of RAB proteins, and here are basically the targets, organelles that they sort of function on. So RAB1 is between ER and Golgi, RAB2, the Golgi network, RAB3A, synaptic vesicles and secretory granules. You don't have to memorize these, just know that the RABs are involved in fusion. Okay. And so here's just another molecular mechanism that I want you guys to sort of appreciate. Just like with vesicle formation, I want you to understand sort of the targeting and fusion of these vesicles. Okay, so here's just a small vesicle. It's got your cargo receptor with your cargo, and the vesicles also have these proteins on them called V snares or vesicle snares. Okay, and so once the GDP has been exchanged for GTP on the RAB protein, it associates with the membrane. Okay, and this helps guide it to a RAB effector protein on the target membrane or the target compartment. So the RAB will associate with a RAB effector protein. Okay, and this is called the tethering step. Now you have the vesicle actually tethered to the appropriate target membrane. Okay, the RAB effector can then bring the vesicle in to what is called sort of the docking state. And during the docking state, you get the formation of what is called a trans-snare pair. Okay, and that's where your vesicle snare is become associated with the target snare or your T snare. So you have a V snare coming together with T snares. And these form coiled coils like we've seen before. Okay, and then once you get these coiled coils that are formed, what ends up happening is the vesicle can sort of pull itself together, and then it fuses, and then all of a sudden the cargo can be released from the cargo receptor. And oftentimes small pH differences between the different compartments, like between the endoplasmic reticulum and between the Golgi, allows this to happen, and this cargo receptor will actually then get recycled back to its donor membrane. Okay, as will the necessary V snares. Okay, so there's a whole recycling thing. So vesicles are moving forward and backwards at the same time. But once again, know how fusion happens, just as a molecular mechanism. Okay, so here's just more detail on that. So normally you have your RAB GDP, oftentimes associated with GDI, the GDP dissociation inhibitor. But once you get vesicle fusing, the RAB-GEF will exchange the GDP for GTP, and the RAB protein then gets sort of embedded in the membrane. You can also have things like PI3 kinase phosphorylating the phosphoinositides. And then that helps recruit this little RAB effector domain where the RAB effectors are, as well as the RAB proteins. Okay, and then these are sort of waiting as sort of a site for the vesicle to fuse. Okay, and once the RAB effectors pull the vesicle in, you get formation of the trans snare, where you have the V snare associated with a T snare in this trans snare pairing, or this trans-snare complex. Okay, and these are all sort of held together by coiled domains. And to actually get the fusion, once again, you have a hydrophilic surface coming together with a hydrophilic surface, and these are both, this is sort of a thermodynamically unfavorable event, and so what ends up happening is the trans-snare pairing sort of pushes things out, pushes the water, and then it sort of pulls the membranes together, and what you get is sort of this sort of stalk formation and then hemifusion, and then once you get sort of the hemifusion, it can start to spread, and then ultimately you get fusion. So just sort of consider the energy requirements here with this trans-snare pair sort of pushing these sort of two membranes with their hydrophilic domains together, right, and pushing them apart, and then having the hydrophobic domains sort of pull apart as well. Okay, a lot of energy is required for that, and it's all sort of mediated by the sort of the squeezing of the trans-snare. Now, once you have fusion, so you have your docking and then your fusion, you're stuck with this very tightly assembled trans-snare pair complex, and the cell wants to actually recycle the vesicle snare or the V snare. Okay, and so what it does to do that is it uses this protein called NSF, and NSF and accessory proteins use ATP to break the trans-snare pair, and then this snare can actually be recycled back to its donor compartment, whereas these sort of just stick around. Okay, so NSF is going to use ATP to dissociate that. So this vesicle fusion requires a lot of energy, but the energy is inherent in the system, and what you need is the energy of ATP to actually break the trans-snare pair apart. Okay. Okay, so now we're going to go on to the viral entry, which is sort of very similar to vesicle fusion. And I mentioned in the last lecture that I wanted to talk a little bit about viral entry, and also in what we're sort of experiencing now with COVID-19. I think it would be a good opportunity to sort of really appreciate this. And you can all tell your folks, if you're home at the dinner table tonight, how you learned about how COVID-19 invades. But in this, the example your book uses is actually using HIV, and here's just sort of an image of a viral particle, and it's actually sort of fusing with the plasma membrane, and here you can see the fusion of it with the plasma membrane. Okay, and viral entry is very analogous to the process of vesicle fusion, where you sort of are seeing this happening. Okay, where a vesicle comes in and it fuses. Okay, but it doesn't use the trans-snare pair. It actually has oftentimes it on the coat of the virus. Okay, so here is your HIV protein, your HIV virus. Here it has an outer membrane, and here is sort of the nucleocapsid of the virus. And what you have are these HIV fusion proteins. Okay, and they'll bind to a receptor on the surface of a cell, GP120, which is then associated with CD4, and that is then brought to a chemokine receptor, and then eventually the nucleocapsid and the virus will insert into the membrane, and the fusion protein comes together to sort of form a system that's analogous to the trans-snare pair. Okay, and it pulls it together, and it causes the membrane of the virus to fuse with the plasma membrane of the cell, and the nucleocapsid is then released into the cell. In the case with HIV, it's a retrovirus, so the RNA in the virus will actually then be converted to DNA by reverse transcriptase, and then that DNA will get integrated into your genome. Okay, so you will have HIV for the rest of your life if you ever get an HIV infection, or people who get HIV infections have HIV for their entire lives, because it's actually integrated into their genome.